Today, I will define a new semantic similarity measure that relies on information content and semantics to determine relateness of Go annotated entities. This is a joint work with Ignacio Traverso Rebon. To motivate our work, let's consider the result of the Cleopatra project, which suggests that the combination of two monoclonal drugs in a chemotherapy treatment increases the median survival to nearly 5 years for patients with metastatic breast cancer. So the question is, how computational tools can be used to determine new interactions between drugs and targets? In this particular case, when we analyze the drugs, we can see that these drugs belong to the same family, the family of monoclonal antibody drugs. Also, the targets or proteins are of the same family, the hair family of receptors. In this case, we may say that both drugs and targets are similar. Making use of the similarity values, the prediction hypothesis can be applied and deduce in this way new interactions between drugs and targets. Our problem is how to accurately compute similarity values between genes, exploiting all the knowledge encoded in the gene ontology based on annotations of these genes. Ontologies encode different types of knowledge, for example, the taxonomic knowledge. Analyzing the taxonomic of an ontology allows for determining how similar are two terms based on their lowest common ancestor or their specificity according to the path length to the root of the ontology. Furthermore, ontologies represent different types of relations, regulates, part of, has part, and negatively and positively regulates. And the semantics of these relations can be represented using ontology rules. So for example, in this case, an implicit instance of the part of relation can be inferred if a relation of is explicitly stated and also an explicit instance of the relation part of. Additionally, two instances of the relation regulates can be inferred because there is one instance of the positively regulates, another of the negative regulates, and two instances of the relation is A. Finally, information content allows for estimating how informative is a Go term in a data set of annotated genes. Based on these three types of knowledge, our research question is how the combination of the taxonomic knowledge, the semantics encoded in the ontology using the rules, and the information content can be used to improve the accuracy of the estimations of relateness and semantic similarity of two genes annotated with the gene ontology. In our agenda today, we will briefly analyze the related work. We will present our approach IC-OnSim, the experimental results of our empirical study, the conclusions, and future work. The problem of determining relateness between two annotated concepts have been extensively studied in the literature, and several semantic similarity measures have been defined. For example, the taxonomic, the path and neighborhood similarities, the annotation base, and the information content. To illustrate the main properties of these state-of-the-art approaches, we use a portion of the gene ontology, where solid arrows represent explicit relations, dash arrows represent implicit relations, and NR stands for negatively regulates and RG for regulates. Taxonomic-based similarity measures compute similarity based on the structure of the ontology. They basically consider the is-R relationship, the distance to the lowest common ancestor, and the distance to the root of the ontology. The rest of the relations or the semantics of these relations are not considered during the computation of the similarity. The path-based similarity measures compute similarity based on the number of paths that connect two ontology terms. Similar than before, these similarity measures do not distinguish between the different types of relations or to the semantics of these relations. Finally, the ontology annotation-based similarity measures estimate the relateness depending on the similarity of the annotations that are used to describe the annotated entities. The annotations are usually compared based on taxonomic similarity measures. Our approach is IC-OnSim, and the objective of our approach is to overcome the limitations of existing similarity measures. The idea is to combine taxonomic knowledge with knowledge encoded in the neighborhood of two Go terms and the information content in order to have a more precise value of. As annotated-based similarity measures, we distinguish between the Go annotated entities and the Go terms. Specifically, Go terms are used to build a bipartite graph, where edges are labeled with the IC-OnSim value of the corresponding Go terms. From the bipartite graph, we compute a one-to-one weighted perfect matching using the Hungarian algorithm, and weights are the corresponding IC-OnSim values. Thus, IC-OnSim receives two Go annotated entities and produce a value of semantic similarity. The taxonomic similarity is also received as input, and the semantic of the neighborhood of the compared Go terms is considered during the computation. Finally, information content of the Go terms is also taken into account. To compute the inferred neighborhood represented with the dash arrows, we use all the rules of ontology to justify how this inferred neighborhood is computed. Also, we consider all the relations between the object properties. For example, one relation is the sub-property relationship between negatively regulates and positively regulates and regulates. To represent the justification of an implicit relation, we have a special data structure that keep information about the origin of the relation, the target of the relation, and the justification. So, for example, to represent that there is an implicit relation between A and C, and this relation is regulates, we will have an instance of this tuple where we have that A is the origin, C is the target, the relation is regulate, and the justification is that A is a B, B negatively regulates C, and negatively regulates is a subproperty of regulates. Further, we can represent that the relation between EC is implicit, and this relation is represented using the tuple EC regulates and the justification that E is a D and D regulates C. To compare how similar are two implicit relations, we build a bipartite graph with the justification of these two implicit relations. So for example, in this case, we compare these implicit relations by comparing the justifications, and we build a one-to-one perfect matching. And from this one-to-one perfect matching, we compute an aggregated value that indicates how similar are two implicit relations. Once the neighborhood is computed, information content is measured in terms of the frequency of the goal term and the number of annotated genes in the data set. MICA is computed based on the common ancestors of two goal terms and correspond to the common ancestor with the highest information content. Finally, the IC formula and the MICA goal term is used to compute the similarity in terms of information content proposed by ResNet. This is normalized by the maximum value of information content in the data set. The IC-OnSim architecture is presented here, and as we can observe, we receive two genes annotated with the gene ontology, and combining the taxonomic similarity measure, the information content measure that we just defined, and the neighborhood similarity measure. These three values of similarity are combined to build a bipartite graph between the Go annotations of two Go annotated genes. And from this bipartite graph, the one-to-one weighted perfect matching is computed. From this one-to-one weighted bipartite matching, the formula presented here is applied. Basically, we sum up the values of similarity in the edges of the one-to-one matching and normalize that values for the maximum cardinality of the annotations. We empirically evaluate our approach with the collaborative evaluation of semantic similarity measure tools. This tool provides two datasets of proteins, the 2008 and 2014. We studied the impact of considering semantics and information content. The annotations were retrieved from the Goa UniProt and separated into the Goa hierarchies. We consider three baseline measures: the normalized sequence similarity, P-Find, and the enzyme comparison, ECC, and we report on the Pearson correlation. Hermit is the out-reasoner of our approach. Furthermore, we evaluate IC-OnSim with respect to 11 state-of-the-art semantic similarity measures. We compared the values of the sequence similarity represented in this black solid line with respect to the 11 semantic similarity measures and IC-OnSim. First, to understand the impact of just considering taxonomic knowledge, we report on unseen tags. Unseen tags uses the annotation-based similarity but only consider the taxonomic knowledge. And we can see the correlation with the sequence similarity is relatively low, and this measure assign high values of similarity to pairs of proteins that are dissimilar according to sex-SIN. On the other hand, when IC-OnSim is used to compute the similarity, we can observe that dissimilar pair of proteins receive low values of IC-OnSim similarity. And the Spearman correlation of IC-OnSim with respect to sex-SIN is quite high. When we compare IC-OnSim with the rest of the semantic similarities and with respect to the baseline, we can observe also that performs quite well. Similarly, when we study the performance of unseen tags in the 2000 dataset, we can observe that as before, unseen tags assign high values of similarity to dissimilar pair of proteins, and the correlation is not so high. But when we consider IC-OnSim, we could see that IC-OnSim is able to assign very low values of similarity to dissimilar pair of proteins, and the Spearman correlation value is quite high. And as before, when IC-OnSim is compared in terms of Spearman correlation with respect to the baseline measures, we exhibit a very good performance. To conclude, we have presented a new semantic similarity measure that is able to combine the taxonomic information encoded in the ontology, the semantic of the neighborhood, and the information content to produce a value of similarity of Go annotated genes. Our experimental results also suggest that our approach highly correlates with respect to the baseline similarity measures. But the performance of IC-OnSim as other annotation-based similarity measures depends on the quality of the annotations. So it's very important that experts curate these annotations in order to obtain precise values of these measures. In the future, we plan to apply IC-OnSim to other domains. For example, currently we are using this semantic similarity to decide how similar are two surgeries that have been annotated with ontologies. Also, we are defining new algorithms in order to provide more efficient implementations of IC-OnSim. This is all for today. Many thanks for your attention.